In the 18th century, chemists could synthesize acids, salts, and solvents, but never anything alive. When they looked at living matter, like muscle or leaf tissue, they found complex substances that didn't behave like anything from the lab. And that gap fed a powerful belief that life depended on a vital force, something beyond chemistry and physics. Yet this belief wasn't just mystical, it was a serious, structured theory. And for decades, nobody could prove it wrong. Vitalism held that organic compounds, the building blocks of life, couldn't be made without this elusive life force. This idea became especially entrenched as chemists cataloged more and more compounds from plants and animals that couldn't be created in a lab. But in 1828, a young German chemist named Friedrich Wöhler stumbled into something strange. He was trying to make ammonium cyanate, an inorganic salt. Instead, he ended up with urea, an organic compound found in urine, long thought to be producible only by the body. Wöhler wrote to a colleague, "I must tell you that I can make urea without the help of a kidney." His experiment cracked open a door, but it didn't slam it shut. Many chemists dismissed it as a fluke. After all, urea was a relatively simple molecule. The mystery of life, they argued, still lay beyond synthetic reach. But Wöhler's work inspired others. Hermann Kolbe, another German chemist, went further in the 1840s. He managed to synthesize acetic acid, the main component of vinegar, from carbon disulfide, a completely inorganic substance. That was harder to explain away. And by the late 19th century, Emil Fischer was unraveling the structures of sugars and purines, showing that even the chemistry of genetic material followed known principles. Each new synthesis, each organic molecule coaxed into being from lifeless ingredients, chipped away at the need for a vital force. Still, resistance lingered. Vitalism adapted. Some researchers conceded that simple organic compounds might be made synthetically, but insisted that complex biological processes, like fermentation, still needed a living catalyst. This was the line that French chemist Louis Pasteur took in the 1850s. He showed that fermentation didn't occur in dead yeast extract, only in live cells. To him, that supported vitalism. The process was tied to the living organism. But in the 1890s, German chemist Edward Buchner repeated the experiment with more refined methods. He crushed yeast, filtered out the cells, and added sugar. Fermentation still occurred. There was no living cell, only enzymes, proteins that could drive the reaction on their own. Life, it seemed, could be reduced to mechanisms. The collapse of vitalism wasn't sudden or complete, but over time, its core claim that living matter obeyed different rules eroded under experimental pressure. Today, no serious biologist invokes a vital force. We can sequence genomes, engineer bacteria, and model protein folding with computers. But that doesn't mean the full complexity of life is solved. Biological systems still behave in ways we can't always predict. Emergence, feedback, development. These are not vital forces, but they still stretch our tools and understanding. So we return to that early conviction that life must be fundamentally different from matter. The modern answer isn't yes or no, but rather, life is matter, organized in ways that create new rules. The laws of chemistry don't change, but how they play out in cells still holds mysteries. Vitalism is gone, but the urge to find something special in life? That's harder to erase.